yeah, I'm a little scattered at the moment. I apologize. Okay, so, okay, I have this thing blocking me. Hold on, guys. I'm sorry. Let me get my head into gear. Okay, so last time we started talking about the RAS system and ways that we control blood pressure. So we talked about how first the kidney has these intrinsic ways that it can regulate blood pressure. And the issue with blood pressure is that blood pressure will change the filtration rate. So the kidney has the glomerulus, it's this filter, and it wants to maintain a constant GFR, it's a constant glomerular filtration rate, a constant rate of things passing through the kidney. However, if we increase the blood pressure, if we increase the force pushing on things from the afferent arteriole into the glomerulus, we might increase that filtration rate. And so the kidney has intrinsic ways of regulating that to maintain a constant GFR when it is just a little change. However, when it becomes a very big change, this is where the RAS system kicks in to try to regulate a large drop in blood pressure. Okay, so I rearranged a few things to go back over some key points before moving forward because it's complicated. There are a lot of moving pieces, and I just want to reinforce some of this material. I also want you guys to draw, that was that activity due today, right? Draw out this system, make it colorful, make it big, do a version in pencil, erase it, fix it, do it again. Really try and see how all these pieces come together with the same goal of increasing blood pressure. So again, we're looking at those juxtaglomerular cells there, and they're going to release renin with the end goal of increasing blood pressure. And they can be triggered by three different things. They can be triggered by their own ability to sense low blood pressure, by activation of our sympathetic nervous system. So in stressful times, we're going to increase that BP. And also by those nearby macula densa cells in the distal tubule that will sense the filtrate, what's happening in the filtrate. So they can be sensed by what's happening in the arterial, what's happening in the filtrate, or an input from our sympathetic nervous system. And what renin does overall, and we'll go into this a little bit more specifically, is it activates angiotensinogen, which is released by the liver, turning it into angiotensin 1, which is then cleaved by ACE to turn into angiotensin 2. And angiotensin 2 has a wide array of effects. It causes vasoconstriction by itself. It tells the kidneys to reabsorb more salt by itself. And remember, when you're thinking about what is the relationship between sodium and water, the sodium, if wherever the sodium goes, water is going to follow. Water, because of that osmolarity, right? Water is going to move to try to decrease the osmolarity, trying wherever the saltiness is, it's going to go to the salt. So if we reabsorb more salt, water will then follow. And when water follows into the blood, we increase blood volume, we increase BP, blood pressure. So you need to keep that in mind. So angiotensin 2 will vasoconstrict. It'll have the kidneys reabsorb more salt, which will increase our water reabsorption, increase our blood volume, increase our BP. And it will also have effects through the hormones it signals. So it's going to signal to the pituitary to release antidiuretic hormone, also referred to as vasopressin, and for the adrenal glands to release aldosterone. And what are these? We spent some time last class talking about what these hormones do. So antidiuretic hormone, or vasopressin, is released from the posterior pituitary because of signals sent to the hypothalamus. So hypothalamus in our brain and the posterior pituitary, they're connected, they're linked to each other. And so the hypothalamus will get a trigger, either what we talked about, angiotensin 2, or the hypothalamus has its own osmoreceptors that will sense the hyperosmolarity of the blood and say, like, we need more water in, we need some more blood volume in here. They also get signals from atrial receptors that are saying, like, BP is falling, or sympathetic nervous system, which says, I'm really stressed, I need a fight or flight response. All of that will cause the hypothalamus to tell the posterior pituitary, okay, dump it, release the ADH, release that vasopressin. The vasopressin has two main actions. One, it will cause blood vessel constriction itself, and more importantly, it will increase the fluid reabsorption in the kidneys. And this is through increasing the amount of aquaporins on the membranes in the collecting duct. And this is what we talked about last time. So vasopressin, or ADH, antidiuretic hormone, binds to its G protein coupled receptor. This signals through adenylyl cyclase to cyclic AMP, which activates protein kinase A, which then phosphorylates these little vesicles that are primed, they're ready to go, they're full of aquaporins, to go to the membrane, to fuse, and to fill the space with aquaporins so that water can go. And this is really fast because it doesn't have to wait for transcription. It's just a fusion of a membrane vesicle into a membrane. Aldosterone, on the other hand, is much slower. So this is a mineral corticoid secreted by the adrenal cortex, which will then cause the kidneys to increase sodium reabsorption, which water will follow, as well as increase our excretion of potassium. And it can be triggered, again, by angiotensin 2, as well as by high plasma concentrations in the blood, which will be a problem for your body, and your body will say, we got to get rid of this potassium. We should switch it out for sodium. We should do that exchange that occurs at the ATPase. These will target, again, the end of the line, the distal tubule, the collecting duct, and because it's a steroid hormone, it can squeeze through the plasma membrane. This is what we showed last time. Again, aldosterone wiggling on through that plasma membrane, going to the nucleus, turning on transcription for target genes. And the target genes here are that sodium potassium ATPase in the basolateral membrane of the principal cells and the distal tubule and the collecting duct. And so what that will do is it will take more potassium out of the blood and put it into the filtrate, and the reverse for sodium, take it from the filtrate, put it into the blood. It also increases the expression of potassium and sodium channels to allow for this movement through the epithelial cells. So again, the ATPase generates the gradient, and the channels open, and the ions will flow down their gradient that's generated by the pump. This is a net exchange of plasma potassium for sodium in the urine. So we're going to take sodium and put it into the blood, and take potassium from the blood and put it in the urine. And again, it doesn't directly affect water. Water will just follow the sodium. So if we compare them side by side, ADH makes our membranes more permeable to water. So what this does is it decreases our osmolarity directly. So it's increasing volume. Remember, osmolarity is the amount of salt you have in a certain volume. And so if we decrease the volume, we're going to be changing the osmolarity. Whereas aldosterone, it plays with salts and water, and it doesn't have a direct effect on water. So aldosterone is going to increase sodium reabsorption and potassium secretion with no direct effect on water, whereas ADH affects the water. The net result is similar, but they have different targets, and so that's important to keep in mind. Okay, so if we go back, that was like the end of the pathway. So what we talked about is the second A, the aldosterone/ADH part here of the renin-angiotensin-aldosterone system. End of the line. Let's go back to the beginning of the line a little bit and talk about what happens here. So juxtaglomerular cells are going to secrete renin, which is an enzyme, which will convert angiotensinogen to angiotensin 1 in plasma. And then the blood vessel epithelials will secrete another enzyme that will convert that to angiotensin 2, called ACE. All of this is happening, the signal for this, again, is happening in this juxtaglomerular apparatus. So again, we have that afferent arteriole coming into the glomerulus. We have these juxtaglomerular cells, which are sensing what's happening here in this arteriole. And they're right next to the distal tubule with those macula densa cells, sensing what's happening in the filtrate. All of them working together. You also have, and it's not shown on this picture, some like sympathetic nerves coming in, having an effect. Okay. What triggers the release of renin specifically? So renin is the enzyme released by the juxtaglomerular cells when they either sense low BP in the afferent arteriole, when they get told by a sympathetic nerve, hey, I'm stressed. Okay, let's do it. Let's raise our blood pressure. Or when the macula densa cells sense issues in the filtrate, they're going to release prostaglandins, and because of that short distance they're traveling, we call that a paracrine signaling. Okay, this is again showing you how we get renin to be released. So we either have that drop in blood pressure. It's going to tell our afferent, it's going to be sensed in the afferent arteriole, which the juxtaglomerular cells will say, hey, we should release renin. We have changes to the filtration rate, which will be sensed by the macula densa. Hey, filtrate is wrong, we will release renin. Or we have that sympathetic activity, which will tell the juxtaglomerular cells, hey, we got to fix this. They will release renin. So 3 ways renin can be released from the juxtaglomerular cells. Again, it's going to move through the pathway. The angiotensin has those diverse effects. It will stimulate sodium reabsorption from the proximal tubule itself. It will vasoconstrict blood vessels after in the postglomerular blood vessels, and it will again cause the release of aldosterone and ADH, which have longer lasting effects. Angiotensin 2 itself, it vasoconstricts, which will increase BP. It causes aldosterone to be secreted, which affects the sodium and the potassium levels, which will then cause a change in pressure. Goes to the posterior pituitary, right? We're going to release ADH. That's going to have an effect of water reabsorption. And then angiotensin 2, again, can also tell the hypothalamus to increase our thirst. So we'll drink water ourselves to increase our blood volume. This is what it looks like in a different way. So keep in mind the effects of renin and angiotensin are happening in blood vessels. They're hormones, so they're going to be secreted into circulation. So angiotensinogen comes from our liver into circulation. The juxtaglomerular cells are going to release that enzyme renin, which will cleave angiotensinogen into angiotensin 1 into a blood vessel. ACE is released from the capillaries themselves. A lot of it is released in the lungs, but it's released in all capillaries. And it's going to convert angiotensin 1 to angiotensin 2 in the vessel. And that will then go and signal to the adrenal cortex to release aldosterone. Okay. So this is the same thing in another way. I'm showing you the same information in multiple ways because 1, repetition is important, and 2, everyone has a different learning style. So I'm trying to portray complex information in a few different ways. But I think the best thing you could do is to try to synthesize all of this, to try to put all this information together and make your own diagram. But I'm giving you some examples. Okay, so if we have that drop in blood pressure, renin is going to be released from the kidney. We know what cells are involved with that, right? Angiotensinogen will be then converted to angiotensin 1. ACE will be released. It says here from the lungs. It's now understood that ACE is released from capillaries in all parts of the body. The largest contribution is from the lungs. And it will be converted to angiotensin 2, which will go and signal to the adrenal gland to release aldosterone. It will also cause vasoconstriction on its own. Okay, so put it all together, right? We got, again, angiotensinogen to angiotensin 1 to angiotensin 2, started by renin here. It's going to have sympathetic effects. It's going to affect the kidney. It's going to go to the adrenal gland and cause aldosterone to be secreted. It's going to go to the pituitary gland, cause ADH to be secreted. It's going to vasoconstrict on its own. All of this working together is going to increase our water and salt retention. So it's a complicated system that has the same goal of bringing back up our blood volume, which brings up our blood pressure. When that blood pressure gets high, the system will turn off. Let's talk about another layer of control here. So that system, the RAS system, brings BP up, but you also have a system in place that will bring BP down. So natriuretic peptides regulate the salt balance too, and they favor the appearance of salt in the urine. So rather than increasing salt and water in the plasma, they're going to increase salt and water in the urine. And so we have 3 different kinds. We have atrial, AMP, brain, BNP, and C-type, CNP. And we're going to focus our attention on atrial, ANP. So AMP is produced by the atrial cells in response to stretch. So high blood pressure, high blood volume, if it gets to be too high, will activate these stretch receptors, and the atrial cells will make AMP and release it into circulation. And they will go to the kidney, and they will increase, they'll work through cyclic GMP signaling, as opposed to cyclic AMP signaling, and they will increase sodium excretion. So they're going to also increase blood flow by relaxing the mesangial cells, so relaxing the filter, creating a higher filtration rate, and they inhibit the release of renin, and they inhibit the release of aldosterone. So it's the way of turning the whole system off. looks like. Again, if we have high blood volume, that's going to cause stretch in our atria. ANP will be secreted, and it's going to have all these different effects. It's going to dilate the afferent arteriole, which is going to increase our GFR. They're going to have a decrease in sodium reabsorption, which means more of it will be excreted. We'll turn down renin, and then that will in fact turn down aldosterone signaling. Okay. And this is another diagram putting it all together. The different cues that cause aldosterone to be secreted, as well as the contribution of ANP. I'm not sure that this is a particularly helpful diagram, but I included it if this type of visual speaks to you better. I liked that it included ANP into the diagram, and it was simpler than the diagram provided by your textbook, which I'm going to show you, and I'm not going to go over because it's just a little frightening. I'll show you at the end. Okay, but what I next want to talk about is this last component of the system, which is the hypothalamus. So your hypothalamus regulates thirst too. So not only does your hypothalamus respond to angiotensin II, and it responds to osmoreceptors to help signal to the pituitary to release hormones, but the hypothalamus will actually increase your thirstiness, which will allow you or tell you it's time to drink water. And because water consumption will affect that water balance as well. So the hypothalamus is that a part in your brain that has an incomplete blood-brain barrier, and so it can actually detect what's happening in the plasma. And so these osmoreceptors not only will a signal there tell the hypothalamus, okay, tell the pituitary, release hormones, we got to get that pathway going with ADH, but it will also send the signal to your brain saying, gee, I'm thirsty. It's time to drink. Okay. This is the confusing picture that your book puts here, and I included it for completeness. I'm not going to go over it because it's just a lot. I think it's just a lot. But if you like a lot, here you go. Here is a lot. Okay. So what we did today is we finished going over the RAS system. You should now, that's scary. It is scary. It's, yeah. I think some people's brains work this way. I'm a word person. I find like a block of text to be the best way for me to learn things. And I think visual people like more visual than this. Like this is a mix between words and visual with not enough of either, in my opinion. But it's here. This is how your book shows it to you. I think that like this one is a little bit better. It's like virtually the same, but you can at least see the organs and stuff. Whereas this, yeah, I'm just gonna leave that. So today we finished the RAS system. You should now feel comfortable drawing your own diagram. It doesn't have to look like any particular one, but try to be inclusive of all the components and what everything does, and really work on like synthesizing that information. Again, apologize for my Friday scatteredness. Yeah, it's been a week. It's been a week, and hopefully Monday will be better. You guys have that paper due. Second half of the alphabet people, I'm grading yours. First half of the alphabet people, Dr. Pogue is grading yours. If you still have questions, direct it to your relevant grader. Hopefully the comments that were made on your draft were helpful so that you can polish that up. And then next week you've got that last lab, and I posted some information, I believe, about your last exam, Unit 4 exam. So a note about the date for Unit 4 exam, it's May 1st. Technically, that is not a school day. The calendar is supposed to end on that Thursday being the last day of classes, but I put your exam on that Friday as opposed to during lab on Thursday, because in case we go over any new material on Wednesday, I didn't want you to just have like a 24-hour turnaround to learn it. I wanted to give you like a 48-hour turnaround to learn it. I've already had one student contact me about a conflict with that time. If you also have a conflict with that time, please let me know, and we can adjust some things, but I think that is the plan I'm going to stick with. Yeah, if you guys have questions, let me know. Otherwise, I will see you Monday.